Hello, my name is Michael Paulini. I work at the WTSI Institute in Cambridge in the UK as part of the grid team, which does assembly, QC, curation, and finishing, as well as the AST team, which is working on aquatic symbiont systems. The AST team is part of the Aquatic Symbiosis Genomics team project. That is a jointly funded project from the WTSI Institute and the Gordon and Timur Foundation, trying to support research communities on aquatic symbiosis systems in marine and freshwater species. So each system usually consists out of one host and one or more symbionts. Their kind of assets range is quite large from one to a lot. The symbionts tend to be microbes in our case, which are bacteria, prokaryotes, eukaryotes, algae, the whole range. We aim currently to do 500 symbiotic relationships, which is kind of for symbiont systems. And originally we estimated it to be 100 species, but it depends on how you count, especially with the bacteria, because we kind of do have quite large systems sometimes. So here at Sanger, we handle anything from sequencing to the final publications. You probably will hear from Victoria and Graham about some of the steps too. But I would just like to go through them again in case you haven't seen them. So kind of we do assembly and sequencing, which is kind of DNA and RNA, as a transcriptome and genome sequencing. We do lots of R&D on top of that, so it's not only hi-fi sequencing. We tried nanopore, we tried optical maps, we tried OmniC. It's a lot of it is part of the R&D going into it for the specific species that because the ASG project is very varied. We tend to not have one protocol that works with all of them, so we are constantly developing new ones. After transcriptome and genome sequencing, basically in assembly, we scaffold it and classify the scaffold. So the idea is to sequence the whole cobion system in one go and classify it afterwards bioinformatically. We run various scaffolding pipelines, salsa, gas, etc. And also our classifier is based on multiple different tools. So kind of with more mainstream ones like Tiara and BTK. And then we also involved in developing FCS genome with NCBI. And based on that one, basically we split the outcoming scaffolds into species beams in a way. These ones will be checked manually in parts for QC and for errors. Fix small problems, and then we will annotate them with repeats and gene models, which is also kind of multiple tools at the moment. So depending on which species it is a breaker or procker at the moment, but we are trialing other things like zebra. And then the final data will be made available. And it's usual suspects like RMSDC, Ensemble, we run UCC assembly hubs, we publish the reads, etc. We also have a portal to show the progress of each species on the portal Aquatic Symbiosis Genomics Project. So I would like to talk about the ones we actually curated so far. So we focused mostly on giant clams, which is the nice one up here. Corals and sponges, giant clams and the corals we did so far have the advantage that it's the same symbiont, it's a protist. Symbiodinium. The sponges we have so far have bacterial symbionts, but they're quite big and quite good to handle so far from our protocols. We curate the assemblies or the scaffolds based on high-C heat maps, which are like physical interaction of bits of the sequence with other bits of other sequences. So basically, the darker the heat map, the more interaction it has, the more interactions, the physical closer it's supposed to be. So you can find things which are the same thing, like a chromosome. You can see things like here, where you have like one sequence which has no physical interaction with something else. It's very likely not from that species, but from a different one. So it could be contamination or cobiont. We find small sharpnel things, which the scaffolder couldn't insert based on yeah, high interactions with other sequences. We can see if it will insert them again. We can find inversions, where basically the sequence is septic. Has lots of interactions at the beginning of the B, so kind of it flips the B, would be good. And what we also see is retained haplotypes. So kind of if you have if you have basically multiple haplotypes present in the same assembly, they tend to be sometimes are joined together into one sequence, which is not intended. So you see basically half coverage over one stretch and half coverage over the other stretch. They tend to be quite closely related because haplotypes of the same sequence. They tend to be joined in repeats and gaps artificially by the scaffolder and need to be pulled out afterwards. We do run an automatic haplotypic duplication purging pipeline, but it doesn't catch all of them. So now the before and after, while we do it actually, that are two giant clams, the three ductors. We have more giant clams, but kind of the nicest ones. So what you can see on the left is the before, on the right afterwards. You can see a lot of the shrapnel bits that have been inserted into the scaffolds now. The scaffolds are also kind of now reduced to the expected chromosome number. Quite often we have actually telomere sequences on both sides of the chromosomes. So it makes actually a quite big difference. So the giant clams were quite nice. So we did a coral, the Cordia of Florida, which is a nice colorful coral from the picture before, which is half the size of the giant clams. But needed a lot more edits. One of the reasons is that one had a lot of haplotypic duplication inside. That was remaining after scaffolding and haplotypic purging, so kind of a lot of manual work went into it. So kind of it's not directly related to how long the assembly is. The manual work involved is quite heavily dependent on the quality of the assembly itself rather than size. We are currently working on diatoms, so there's the tiny ones, with cyanobacteria symbionts. We are also working still on the Symbiodinium cobionts, that we can get them out together as a host. And we have one new coral coming in, and our first sponge. This sponge is the one on the right. And whereas it looks like the high doesn't have too many physical interactions, I'd like to point out that that one is actually a screenshot of the host and the cobion. So quite a lot of the empty lines are from cobion system. We did already start curation a bit, and it does look actually like it will resolve quite nicely. Annotation-wise, we will annotate, or we have a pipeline to annotate repeats and gene models. The gene models will be shown currently on the Contrast Sanger hosted assembly hub, so you can attach genome browsers like the WTSI browser to it and see the data we provide, including RNA-seq alignments. We are talking to ENA and Ensemble because they showed interest in showing it too. We also provide our classification of the scaffolds as blob toolkit viewer, which is publicly available. So it is possible to see which sequences belong to which one. Not necessarily down to metagenomics level, but it's quite helpful for people to see which ones were found. We also will provide, or are providing, genome notes for each of our species, which will describe it to the public. Yeah. And I would like to thank our ESG partner hubs, which provides the samples and which are eagerly waiting for us to provide them with the species of choice. And also people working at Sanger in the ESG project. Thank you. That was it. Cheers.